we will talk about the Human Genome Project. The Human Genome Project, which is also known as the HGP, actually genome implies to the number of genes present in a haploid set of chromosomes. Okay, so the basic things about this project was that it was a mega project, and it was considered a mega project because its estimated base pairs were 3 into 10 to the power 9 that were to be found out. Then the cost of one base pair would have been $3, and it would take $9 billion for the completion of this project. At the time the project was started, there were no softwares available to analyze the data. A parallel science was bioinformatics, which developed to analyze. To deal with ethical, legal and social issues, a special committee was designed to solve such issues. And it was said that it would be completed in 13 years. It was started in 1990, so to complete it in 2003, most of the work was completed in 2003 except one chromosome. Okay, it was initiated by the US Department of Energy and the National Institute of Health. Let me tell you that it was started by James Watson in 1990 and Colin took charge of it in 1993. So the methodology used, then we will talk about the methodology. They started with the two approaches. The DNA, we know that the DNA contains coding and the non-coding part. The coding part is that which produces, which codes for the protein. Non-coding part are the introns, which are not at all necessary. In our DNA, there are coding as well as non-coding part. In approach number 1, it was said that they would sequence only the coding part, and it was termed as expressed sequence tags. In approach number 2, they said that they would sequence the complete DNA including coding and non-coding part, and it was called sequence annotation. It was accepted that the approach number 2 will be followed. The second thing of methodology was that DNA was cut into fragments and then it was sequenced. Before sequencing, it was necessary that it should be cloned. That means there should be a hybrid, or there should be DNA fragments cloned should be there. And for cloning of fragments, host cells and vectors were required. The host they decided was that yeast would be taken and the bacteria will be yeast artificial chromosome. Then the Sanger's method was used for sequencing. How was it sequenced? You see here I have made a flow chart. The DNA was cut into fragments, then it was introduced into the vector. It was introduced into the host cell with the help of a vector. The DNA replicated inside the host cell and then it was sequenced using the Sanger's method of sequencing. Then what were the salient features of this was the methodology. Then salient features of HGP or Human Genome Project, that it contains 3,164.7 million nucleotide bases. The average gene consists 3,000 bases and the largest human gene, dystrophin, has 2.4 million bases. One average gene will contain 3,000 bases, but there is a largest gene, dystrophin, which contains 2.4 million bases. Understood? Then total number of genes that were found out by the project were only 30,000. The function of 50% genes was unknown. That means that were found out by the project were 30,000, and that another 50% of genes, they were unknown. They were the non-coding parts of the DNA. Then there was also a very important thing they found out was that the repeated sequences made a large portion of the genome. That means a sequence, suppose A, T, C, T, G, T was there. This sequence was found at various places. This sequence, the same A, T, G, T, C, T sequence was found at various places in that same genome. Okay, so less than 2% of the genome codes for proteins. That they found out that rest of the genome did not code for protein, only very less of it coded for protein. Chromosome number 1 has maximum number of genes. So it was the longest chromosome, that is 2,968 genes, and chromosome Y, which is a very small chromosome, had least 231 genes. 1.4 million locations have SNP, that is single nucleotide polymorphism, that is multiple forms of the single polymorphism. Okay, multiple forms of one nucleotide. Suppose we find a nucleotide or we find a sequence of nucleotide AATTCCGG, we will find the same base pairing or same type of nucleotide sequence again with only single change in one nucleotide only. At A position, suppose I have taken here that at the second position there is a change. Instead of A there is T, instead of T there is C. Rest of it is same. Okay, so difference in only one nucleotide. So there were 1.4 million locations, such locations were there. Now, such type of differences were seen at 1.4 million locations. Then this project completed in 2006. Actually, it completed in 2003, but that chromosome number 1 was the last chromosome which they found out, and it took 3 years. So, it took 2006. The applications where this human genome project was used to diagnose diseases that were treated, that were related to genes like carcinogenic or some metabolic genes or some metabolic diseases. Then to treat these diseases, rDNA technology was used. Then application of the genome other than human genome. The genome of non-human organisms were also sequenced, like that of bacteria, Drosophila, yeast and rice. This information, that is the non-human genome was used in the field of agriculture, microbes in human welfare, microbes to obtain various products and microbes to obtain energy products. So this was all about the human genome. Talking about the DNA fingerprinting. The DNA fingerprinting is a technique which was given by Alec Jeffrey. So, DNA fingerprinting is a technique of determining nucleotide sequences of certain areas of DNA which are unique to each individual. That means they are not found in all individuals. We will identify that in unique type of sequences which are found in the DNA. So this technique is used nowadays to identify the unique identity or required for solving criminal cases or paternity of a child. We know that 99.9% base pairing sequence is same in all. So I must tell you that only 0.1% of the total genome of that many nucleotides which we have, 3 into 10 to the power 6 nucleotides whatever we have. So it is only 0.1% is different in organisms. In a human being, 99.9% of base pairing sequence is all same in all organisms. Okay, so this 0.1% is only very important. It is only needed for identification of the person. Okay, this the 0.1% helps in identifying a person. The DNA is separated using the density gradient. So we got, when we will get two heaps, it is that I will tell you. We will get two heaps when we use the density gradient method. Okay, so the bulk method, the bulk DNA, which is all the same 99.9%, and the satellite DNA will be the one which is only 0.1%. So by density gradient method, they are different, they are separated. Then the satellite DNA is the one which has a repetitive DNA segments, which we also talked in the salient features of a DNA. Okay, or the human genome. This is classified on this type of satellite DNA is classified as microsatellites and mini satellites. Then on the basis of what are they classified? They are classified on the basis of AT or CG ratio. You can see here I have written AT or CG ratio and length of the sequence. That means the number of AT and CG. What is the ratio to AT and CG found in the that repetitive DNA segments. Okay, and then we will find that how long that repetitive segments are. Then how many times of repetitions are there. That means what the copy number, how many number of times that same segment is repeated, that same part is repeated in that genome. Then other above criteria, on the above criteria, it is divided as micro and mini satellites. Mini satellites are used as VNTRs, the variable number tandem repeats. So these mini satellites were only used for our DNA fingerprinting. Okay, these are important. That is how many times the DNA sequence repeats. That was important. how many times it repeats. That is used for finding out the culprit or the criminal person. The VNTRs are used as radio labeled probes. With the help of this radio labeled probes only it was done. Now VNTRs are used to identify the DNA material. Method of DNA printing is what? First of all, the DNA is extracted from the nuclei of white blood cells or of the spermatozoa or of the hair follicle cells that cling to the root of hairs that have fallen or been pulled out. So at the point wherever the crime has been occurred, there we will find blood samples, or we will find something like blood cells, or we will find spermatozoa, or we may find some root hair, roots of that follicle roots. So from that we will extract the DNA. DNA molecules are first broken with the help of enzyme endonuclease. Okay. So second part is what? First of all, isolation of DNA is done. That means everything else of the cell will be destroyed. Only DNA will be left. Then that DNA will be broken down into fragments with the help of endonuclease. Then the third point is that separation of fragments by gel electrophoresis. In this way, what happens? The fragments are separated according to the size by gel electrophoresis. Okay. Then there is a method, gel electrophoresis. Then the fragments of the particular size having VNTRs are multiplied through PCR techniques. They are treated with alkaline chemicals to split them into single-stranded DNA. So the separation of fragments is done. Then the blotting is done. In the blotting, transferring the fragments into nitrocellulose is done. Then hybridization with radio labeled VNTRs is done. Studying is done. What can we study? One example I will give you. There is an example. Suppose there are two individuals, individual 1 and individual 2, individual A and B. In individual A, the chromosome number 1, which is having a homologous pair also, we will find out the VNTRs. So the VNTRs are 1, 2, 3, 4, 5. That means 5 times one base pair is repeated. So I write here number 5. In the homologous part, it is that 3 are repeated. 3 times it is repeated. So I have written here 3. In the individual B, in chromosome 1 only, 8 times repetitions are there. And in homologous pair, 2 times of repetition is there. In chromosome number, now we will take the second chromosome of that individual 1, and we will find that in second chromosome there are 4 repetitions, and in its homologous part there are 7 repetitions. Similarly, in individual B, the chromosome number 2 contains 6 repetitions and 1 repetition. Then we will take the third chromosome. The chromosome number 3 contains 10 repetitions and its homologous chromosome contains 6 repetitions. Similarly, in individual 2 or individual B, 4 repetitions and 7 repetitions. So how are the samples tallied? Well, now it will be tallied when we separate them using the gel electrophoresis. The gel of the individual A and B will be that the biggest piece, that is which is the more number of time of repetitions has taken, that is the biggest piece or the longest one. Okay, VNTRs according to the length. Longest one, it will take up the biggest piece will be on the uppermost side of that gel electrophoresis, and the smallest piece will be at the lowest side. So after we hybridize it with VNTR sample, we have find out that how we will do this. I have labeled it 12, 11, 10, 9 and all that till 1. So 1 will be here. You will see for the individual A, I have written that there is 10, 7. Here at the 10th position, I have drawn one gel electrophoresis. Okay, one gel. Then there is one colored gel. At 7th position, there is another colored gel. At 6th position, there is another colored gel. At 5th, there is one gel. At 4th position there is a gel, and at the 3rd position there is a gel. Similarly, in chromosome number in the individual B, there are at different places. Now we will take the sample, and from the sample we will find we will find out the samples, the sample which we have got in that crime position where the crime had occurred, and we will test. Then we find that suppose it has at 1 position, at 2nd position, at 4th position, at 6th, at 7th and 8th, different colored gels are there. So we find that this sample matches with the individual B, hence the individual B is found out to be the culprit. This method was used. In this way, human fingerprinting or DNA fingerprinting was useful. So thank you. The chapter ends.